All right, hello everybody. We're going to talk about cell respiration today. As usual, I'll probably talk fairly fast, but you stop this as need be to take your notes. I am hoping that already in class you have seen or worked on the cell respiration overview packet. So this information should be vaguely familiar to you as I'm talking through it. And you also should have listened to photosynthesis, so you should be familiar with the idea of electrons transferring and that kind of thing. So first of all, we're just going to remind you the connection between cell respiration and photosynthesis, that they are both redox reactions and that they are reciprocal. The products of one are the reactants of another and vice versa. And you should be familiar with both reactions. You should be able to write them out or identify if something's wrong with them. Cell respiration is catabolic and exergonic. Remember that photosynthesis was anabolic and endergonic. And what do those words mean again? Remember, with catabolic, we're talking about breaking down molecules to release energy, and exergonic means energy is being released. So in this case, the molecule that's being broken down is glucose, and you can see on the opposite side, you can see that the glucose has been broken down from 6 carbon molecules all put together into 6 carbon dioxide molecules. So the energy is being released, and then the cell is going to take the energy that's being released from those bonds that are in the glucose molecule. It's going to harness that energy to build ATP, and ATP is an endergonic. The building of ATP is an endergonic reaction. So we're coupling cell respiration with the formation of ATP so that we can build ATP molecules that are going to power all of the reactions that happen in our bodies. So let's read through here, get my picture out of the way. There we go. Glucose is broken down. Its electrons and its hydrogens are removed. It is oxidized, and I want you to see the green arrow. Remember, with oxidation, so reduction means that electrons are gained, and that reduces its overall charge. But with oxidation, the electrons are actually being the electrons are being removed. And when that happens, we often see an increase in the number of oxygen molecules. That's where the name oxidation comes from. So if you take a look, just look at the glucose molecule for a moment and notice that in the original glucose molecule, C6H12O6, the ratio of carbons to oxygens is 1:1. There's 6 carbons and there's 6 oxygens. But when we go over to the other side and we look at the 6 carbon dioxide molecules, now we have a 1:2 ratio. There's twice as many oxygens for every 1 carbon atom. And so that's where the name oxidized comes from. So just be aware of that. All right, the second note here, the second bullet point says the energy released by the breakdown of glucose is used to build ATP. We call that coupling. The energy released is exergonic. That's what's happening during cell respiration. But the building of ATP is an endergonic reaction. It needs energy. So we take the energy that's being released during cell respiration and we use that to do the endergonic reaction of building ATP. The oxygen then gains electrons and hydrogen ions, so then it is reduced. Okay, here's our overview. Hopefully my picture's not covering up too much. So we did a photosynthesis overview. We're going to do an overview of cell respiration. Respiration begins with glycolysis in the cytoplasm. So take a look at this diagram and see if you can look at the diagram while I'm talking you through it. So you're starting outside in the cytoplasm where it's kind of that peach color and the green box says glycolysis. Trying to click on it. There we go. If anaerobic respiration is going to happen, it requires fermentation, and that happens in the cytoplasm as well. Anaerobic, remember, means without oxygen. So if oxygen is not present, then the whole process happens out in the cytoplasm, and the mitochondria are never involved. Aerobic respiration in eukaryotes happens in the mitochondria. In prokaryotes, it happens in the cytoplasm or in the folds of the membrane of the prokaryote itself. Remember that prokaryotes, bacteria, are simply the size of mitochondria. They can't have mitochondria in them. So they have all these processes take place in the folds of their own membranes. All right, and then it includes the citric acid cycle and oxidative phosphorylation. Look at the diagram. So you see glycolysis happened out in the cytoplasm, and then it moves into the mitochondria where the citric acid cycle is taking place, and then something called oxidative phosphorylation, which is where we're making most of our ATP. Here it says it makes 30 to 36 ATP. The packet that we had said 38 ATP. You're going to find so many frustrating numbers in this unit, and I just want you to be aware that the reason we don't have a real precise number of ATP, there's many reasons. And let's see if I can explain any of those reasons quickly. It's not a perfectly efficient process. If any of the conditions within the mitochondria or the cell change at any time, it affects how many electrons are made. So I think, if I understand correctly, the ideal, the absolute perfect conditions, it can make up to 38 ATP. But when the scientists really study it, in fact, 38 ATP are never actually made. It's just theoretically how many can be made, but then the cell is never perfect at making all of it. All right, now that was the overview. Now let's get down into the nitty-gritty and what parts you actually need to know. So for all of cell respiration, and this is actually true for photosynthesis, you don't have to memorize the number. You should know the general reaction. You should know the two redox reactions that are reciprocal. You should know that. For most of the rest of it, you need to understand what's happening, but you don't need to memorize the steps. So you should know the word glycolysis. You should vaguely know what's happening in glycolysis. But for the most part, when you're tested on it, I'm going to give you, or the College Board is going to give you a diagram like this. And you need to understand what's happening in the diagram. You don't need to have memorized the diagram. Okay, so let's back up. Glycolysis. This is the actual breaking down of a glucose molecule. It is almost a universal process. It happens in prokaryotes and in eukaryotes. It occurs outside of the mitochondria in the cytoplasm. And it involves the glucose being cut in half, cleaved into two molecules of pyruvate. So there are each of the pyruvate molecules has 3 carbons. So you're trying to keep track of how many carbons there are and where all the carbons are going. We're starting with a 6 carbon. Look at the diagram. We're starting with a 6 carbon compound. We have to use ATP to cut it into 2 3 carbon molecules. And now you see it branch with those 2 3 carbon molecules. So here it says, the bullet point says it requires 2 ATP to do the cleaving, but then it produces, follow the diagram along, it produces 4 ATP. So we say the net total of glycolysis is 2 ATP. We used 2, we made 4, so our total is 2 that we have produced during glycolysis. Remember that we're trying to make like 36 ATP, so that's not very much ATP at this point. Pay also attention to this last bullet point. It says produces 4 ATP via substrate phosphorylation. So remember, the word substrate came up in our unit, in this unit when we were talking about enzymes. Enzymes and the molecules that they attach onto, those molecules are called substrates. So in this case, when ATP is being made, it's actually being made by an enzyme, and they're coming together, and that enzyme is adding a phosphate. So that's where the word phosphorylation comes from. It's adding a phosphate, and it's an enzyme that's doing that. In a minute, we're going to talk about oxidative phosphorylation. It's the same thing. We're making ATP, only this time the energy it's coming from somewhere else. It's coming from the breakdown of glucose, and it's not being formed in the same way. It will make sense when we get there. Just trust me that you need to pay attention to those 2 different words: substrate phosphorylation, oxidative phosphorylation. Okay. This is the second page of the same diagram. 2 NAD plus molecules are reduced. Got to remember what reduced means. It means 2 electrons are going to be added on, and then the protons, the hydrogen ions, always join in. So it says 2 NAD plus are reduced to NADH, and they often say plus H plus. There's another proton there. The NAD plus are electron carrier molecules when they get reduced. And they should be annoyingly familiar because you saw NADP plus in our photosynthesis notes. So all I can tell you is that they're almost the same molecules, but the NADP plus has a phosphorus, maybe phosphate, group attached to it that this one does not. Do you need to know that? Absolutely not. Do you need to know which one is in which process? Yes. So NADP is in photosynthesis, which also starts with a P. So this one is missing the P and it doesn't happen in photosynthesis. That's how I remember it. I don't know if that works for you, but that's what I recommend. Okay, so look at the diagram. Do you see that the NAD pluses are collecting electrons and hydrogens and becoming NADH? So they're being reduced. If oxygen is present, then the pyruvate is transported into the mitochondria to continue on. Some prokaryotes can do cell respiration across their membranes since they don't have mitochondria. We mentioned that already. Not all bacteria can do that though. So some bacteria are only anaerobic, which means that they can never do aerobic cell respiration, and all they ever make is 2 ATP at a time. So just be aware of that. If there is no oxygen present, then I say, because humans can do it, bacteria can do it, yeast can do it, we go into a process that's called fermentation, and that happens in the cytoplasm, and that's if no oxygen is present. So this is like a sidebar. So what we were doing, I wish I could back up really quickly. Cell respiration is supposed to start with glycolysis, then there's the link reaction, then there's Krebs cycle, then there's oxidative phosphorylation. That's what's supposed to happen if oxygen is present. But we're doing a little step back because now we're saying oxygen was not present. If oxygen is not present, instead of going through being transported into the mitochondria and going through all those other processes, we stop at a process that's called fermentation. Fermentation is super wasteful. Most of the energy that's stored in the bonds of the glucose molecule are not harvested. Hardly any ATP gets made, and it's a super wasteful process. The NADH is oxidized, which means it gives up its electrons and it turns back into NADP+. The pyruvate is then reduced, which means it collects the electrons and it's turned into another waste product. There's several things to be aware of. There are multiple different types of fermentation. So right here in this diagram, you can see ethanol fermentation, which is how the alcohol for beer is made. It's also how yeast work when you're making bread rise, for example. You can also see lactic acid fermentation. That's what happens when we're working out and we don't have quite enough oxygen in our bodies to make the ATP that we're supposed to be making as we work out. And that's what makes our muscles burn. There's many more, apparently, different types of fermentation. These are the only two you ever need to learn about when you're at this level of biology. Okay, so it says, the last bullet point here says, the process is reversible so that once oxygen is available, the end product can be fully broken down. So for example, if you look at the lactic acid fermentation diagram, you end with lactate, and that's what causes your muscles to burn when you're exercising and you haven't been breathing well enough for the level of exertion that you've been doing. That lactate, once oxygen is present, it can go backwards through the process and then return back into the mitochondria and go through all of cell respiration. So it's a reversible process so that the energy doesn't get wasted. Okay, so just a couple quick things about ethanol fermentation and lactic acid fermentation. Ethanol fermentation typically happens in yeast. Just as an FYI, what are yeast exactly? They are single-celled organisms, but they're actually considered to be eukaryotes. They are not prokaryotes like bacteria, and they even have their own mitochondria. So yeast are capable of doing regular cell respiration, but in an anaerobic condition, they will do ethanol fermentation and produce an alcohol that's called ethanol, and that's the alcohol that is used in beer making processes. All right, and I'm going to move on. So now that was just a sidebar if no oxygen was present. But if oxygen is present, then we're going to continue. We started outside of the mitochondria and we did glycolysis and we split it into two 3-carbon molecules. Now those are going to be transported into the mitochondria and this process is called the link reaction. In prokaryotic cells, the pyruvate molecule is transported into the mitochondria. In prokaryotes, remember, the folds in the cell membrane are used so they don't have their own mitochondria, just constantly reminding you of that. Carbon dioxide now starts to form. Remember that that's why we're exhaling. We're bringing in the oxygen so that we can do this whole process. But then the waste product from that is carbon dioxide. We have to get rid of carbon dioxide. You all should know this because carbon dioxide, when it mixes with the water of our blood, becomes carbonic acid. We can't change the pH of our blood because that would screw up our enzymes. Oh my gosh, are you pulling all this together, people? That was biochemistry and cells and everything, enzymes all pulled together in one. All right, sorry, I'm rambling. Carbon dioxide is a waste molecule. It gets removed from each pyruvate. So now the pyruvates had 3 carbons, and now you remove an oxygen from them, and then that means that they have only 2 carbons left. So the new molecule is called acetyl coenzyme A, or acetyl-CoA is how they abbreviate it. And then we also see some NAD plus being reduced to form NADH plus H plus. We're going to have a whole buttload, did I say buttload? Of NADH plus H pluses. They're going to become really important in the oxidative phosphorylation step. So those will come up in just a minute. Okay, so this is the link reaction. We're moving from glycolysis into the mitochondria. That's the link reaction. Now we're going to take the acetyl-CoA and it's going to jump into, look at the diagram, the acetyl-CoA. I'm going to get my picture out of the way for a moment. The acetyl-CoA is now going to be entering the citric acid cycle. This is all happening inside of the mitochondria. So take a look at the diagram. The pink box up at the top, that's the link reaction. The pyruvate has come into the mitochondria. A carbon dioxide has been pulled off. So now it's just a 2-carbon molecule. Some NADH was made, and it becomes what's called acetyl-CoA. And that's going to enter our citric acid cycle. We'll look at another diagram of that in just a second. So going back up here, as each bond from the original glucose molecule is broken, this is during the citric acid cycle, the energy is collected by electron carrier molecules called NAD+, we already met NAD+, and a new one that we haven't heard of ever before, and that one's called FAD. It also carries electrons. So take a minute and just walk your way through this diagram. It's incredibly simplified from what is actually happening. I probably should have shown you a diagram of what's actually happening because it would boggle your minds. So sometime you can look it up. Oh, and just as an aside, I hate the name citric acid cycle. It's also called the Krebs cycle, named after the scientist who came up with it. The reason I hate that name is that it sounds so similar to the Calvin cycle, which is what's happening in photosynthesis. They're both C cycles. I get them mixed up. I have to really concentrate on which cycle is which. So you need to think of a word game or something to keep track of which cycle is happening in the mitochondria and which one is happening in the chloroplast. Okay, so just take a look at the diagram. Notice that some carbon dioxide is being removed. Remember, acetyl-CoA was just a 2-carbon molecule. So 2 carbons are released at this point. So we have just released all of the carbon that was stored in the glucose. The glucose was a 6-carbon molecule. We broke it into 2 3-carbon molecules, pyruvate. We lost 1 carbon, get my picture out of the way, right here in the link reaction right there. And now we've lost the other 2 carbons. This is happening twice because there were 2 pyruvate molecules when we originally cleaved the glucose molecule. So carbon dioxide has been released, and that's all the waste product that we're going to get from this. NADHs are forming, ATP, a little bit of ATP. This is not where we're forming most of our ATP. And then FADH2 is forming as well. So let's take a look at the totals. So 2 more carbon dioxides are released. 3 NADHs are formed, 1 FADH2 is formed, and 1 ATP is produced. And the cycle happens 2 times for each glucose molecule. One more time, I'm reminding you, pyruvate, I'm trying to point at it. There we go. Let's see if I can point. Pyruvate is a 3-carbon molecule, and there were 2 of them because we cut the glucose that had 6 carbons into 2 3-carbon molecules. So this process is happening 2 times. All right, so this is the exact same screen, only I changed the diagram just to give you a little bit more detail. Do you need to memorize this? No, but I just want you to see what's happening. So when the acetyl-CoA comes in, it is joining with a 4-carbon molecule. Do you see the 4C right here? No carbon dioxide has been released, so it's still a 4-carbon molecule. So those 2 come together to form a 6-carbon molecule. immediately a carbon dioxide gets released. That makes it a 5-carbon molecule. Then another carbon dioxide gets released, which makes it the 4-carbon molecule that restarts the whole process. So that's why it's a cycle. In the process, we're making NADH a couple of times. We're making 1 ATP. There's the FADH2 that you haven't seen before, and then another NADH. All right, same process. All right, now we're going to get into the last section of cell respiration, which is called oxidative phosphorylation. It's located in the inner mitochondrial membrane. So let's picture that really quickly. So here's a cell. Cell's really big. Mitochondria is much smaller than I'm making it seem, but here's the mitochondria. This is the outside of the mitochondria. And then remember, it's got all these wavy inner membranes. That's called the cristae, wavy inner membrane. That's the cristae. And that's where this diagram is. So study the diagram while I'm talking. So it's located in the inner mitochondrial membrane called the cristae. The NADH and the FADH2, find them on the diagram. Do you see them in the lower left-hand corner? They were carrying electrons and hydrogens, and they're high energy electrons and hydrogens. Now they're going to drop off. It's the electrons that are high energy. They're going to drop off the electrons at this point. They're dropping them off in what's called the electron transport chain. So look down at the bottom. Do you see where it's labeled electron transport chain? There's like a I don't know what that's called. Bracket. There we go. That's what I'm looking for. There's a bracket that labels the electron transport chain. Those purple blobs, those are proteins that are embedded in the cristae of the mitochondria. And let me just say it really quickly, and then we'll come back and get the details. This diagram doesn't show very well what's actually happening here, which is the electrons are high energy when they get dropped off by NADH and FADH2. And with each passing, with each movement from one protein to the next, to the next, to the next, they're losing energy. They're becoming lower and lower and lower energy. And that energy is being harnessed to push the hydrogens. Look at the diagram to see, and I want you to see the hydrogens are getting pushed through. So as the yellow arrows represent the movement of the electrons, so you can see the yellow arrows, the electrons are moving from one protein to the next, and as they're moving, they're losing energy, and that energy is being used to pump the hydrogens across into what's called the intermembrane space. I think that's the lamest name. Like cristae is a cool word, matrix is a cool word, and then intermembrane space. Like, couldn't we have been more creative? But that's what it's called. So what is the intermembrane space? Here's your mitochondria. Here's the cristae. It's in between the cristae and the mitochondria. So I'm in the outside of the mitochondria. So that's called the intermembrane space. So let me read that last bullet point to you. As the electrons move through the proteins in the electron transport chain, those are the 3, 5, 6 purple proteins that are embedded there. Their energy is harnessed to pump the hydrogens into the intermembrane space. So we are producing a hydrogen ion or proton gradient in the intermembrane space, and that's about to be used for something really important. Now, the electrons have been passing through. They've been going from high energy to a little bit lower energy to a little bit lower energy to a little bit lower energy. At the very end is oxygen, and oxygen's ready to catch them like they're baseballs or something. If the oxygen isn't present, there's nobody there to catch those electrons. The whole system backs up like a plug in a drain. It backs up, then the Krebs cycle backs up, then the link reaction backs up, and then we go into fermentation, because none of this can happen if that oxygen isn't present there. So the oxygen catches the electrons when they're all the way down at the end of the electron transport chain. So it says once the electrons have passed all the way through the ETC, electron transport chain, oxygen captures 2 electrons and 2 hydrogens to make water. So there's our production of water. Water is also considered a waste product. The whole purpose of this process is to make ATP. FAD and NAD plus then return back to glycolysis or the citric acid cycle to be reused over and over and over again. Now, just a quick aside, you don't have to memorize this, but it absolutely helps if you study the diagram and understand this. It has to do with the sort of the conformation of NADH and FADH2. NADH drops its electrons off a little bit earlier in the electron transport chain than FADH2 does. Find them. Look at how NADH is dropping its electrons off at protein number Roman numeral 1, and FADH2 is dropping off its electrons at Roman numeral 2. Imagine a mountain, and NADH is dropping it off at the pinnacle of the mountain. FADH is dropping it 3/4 of the way down the mountain. Who cares? All that means is that there's more energy in the electrons that are being dropped off from NADH, and so more protons get pushed across into the intermembrane space from NADH, and so that means ultimately, just go with this, that more ATP gets produced from them. So those are sort of more efficient molecules. FADH2 produces a little less ATP because it doesn't push across as many hydrogen ions. Because its electrons have missed the first protein that's pushing the hydrogen ions across. Okay, enough of that. All right, the last step, and find it on the diagram in those brackets, it says chemiosmosis. The electron transport chain causes a proton or electrochemical gradient in the intermembrane space. The protons then diffuse back into the matrix through ATP synthase. Now, that should be a name that you're familiar with. We saw an ATP synthase, and I'll remind you of it again in just a minute. We saw an ATP synthase in photosynthesis. The thylakoid membranes also have ATP synthase, and they make a little bit of ATP during the process of photosynthesis. So that's why it looks familiar to you. The free energy that is released as the hydrogen ions move through that ATP synthase is used to form ATP from ADP and inorganic phosphate. This part, the whole electron transport chain and chemiosmosis, is called oxidative phosphorylation. Remember that we are oxidizing glucose. What does that mean again, KJ? It means we're pulling electrons off of it. We're increasing the number of oxygens actually that are attached. You don't need to follow that part, but you need to know they're removing electrons. And then those electrons that are being removed are being used to power the formation of ATP from ADP and inorganic phosphate. So that's called oxidative phosphorylation, that whole process. To phosphorylate means to add a phosphorus. All right, and then, sorry, I'll back up. Chemiosmosis is the movement. Remember, osmosis is the movement of water across a membrane. Chemiosmosis is the name they use when they're showing hydrogen ions moving from an area of high concentration to an area of low concentration. All right, that's it. That's all of cell respiration. We still have some slides that we need to talk about to give you a little bit more information about it, but that's the process of cell respiration. So a couple of things that I want you to see. I love this diagram because I think it's really helpful to show the similarities. I mentioned that you've seen ATP synthase before. So just take a look. Mitochondria on the left, chloroplast on the right. The dark area in the diagram below, it's the intermembrane space in the mitochondria. It's the thylakoid space in the chloroplast. The membrane that you see, the lipid bilayer that you see, that's the cristae, the inner membrane of the mitochondria, or it's the thylakoid membrane in the chloroplast. And then the liquid where the ATP is being formed, that's the matrix of the mitochondria or the stroma of the chloroplast. Okay, so I'll get my picture out of the way again. And both mitochondria and chloroplasts have electron transport chains that produce a proton gradient that is used in ATP synthase. It's called photophosphorylation in the chloroplast because the energy source in that case is light. Remember, light ejected the electrons, and then the electrons were captured in this electron transport chain, and it was used to make some ATP. In the mitochondria, it's called oxidative phosphorylation because this time the energy had been stored by the chloroplast, actually, had been stored in glucose molecules. So now we're breaking the bonds of the glucose molecules. We're removing the electrons. We're oxidizing glucose. And that's what's powering the formation of ATP. So that's what's phosphorylating ATP. So photophosphorylation in a plant, that makes sense, right? Plants need light. Oxidative phosphorylation in the mitochondria. All right, and then this was kind of a cool video. I tried to make it a little bit faster. This comes from a website called BioNinja, and I thought they did a really good job. So they're labeling everything. You see the NADH, it's going to drop off its electron and its hydrogen. So the electrons, and I like that this shows it going downhill better than the other diagram did. The electron is moving downhill through the electron transport chain, and as it loses energy, the hydrogen ions are being pumped across. Now chemiosmosis is happening. The hydrogen ions are coming back through, and the ATP synthase is adding a phosphate onto an ADP to make ATP. We'll do it one more time. That hydrogen ion comes through and allows ATP to be formed. And now that electron is just sitting there. So the electron has to be caught by our terminal electron acceptor, which is oxygen. And some of the hydrogens that have come back through now combine with the oxygen to make water. And that's our waste product. I think that's a good video to kind of help you visualize what's going on. All right, a couple other just things to be aware of. We talked about the coupling of cell respiration with the formation. So cell respiration is an exergonic reaction that releases energy. We couple that with the endergonic reaction of forming ATP. ATP needs energy to be built. In order to build ATP, you need energy. So we put them together. However, we can separate those two processes in organisms that produce their own heat. We're called endotherms. That's as opposed to something like an insect or what am I trying to say? Like a reptile. They're called ectotherms, and they have to get warm by being out in the sun or by being somewhere that is warm. We generate our own heat. How is it that we generate our own heat? We generate our own heat by uncoupling cell respiration sometimes. So it says, in cell respiration, uncoupling oxidative phosphorylation from electron transport generates heat. This heat can be used by endothermic organisms to regulate body temperatures. So please take a look at the diagram. Normally, hydrogen ions can only return, so notice the hydrogen ions are getting pumped into the intermembrane space. Normally, they can only return through ATP synthase. That energy is captured and used to make ATP. However, in some cells, especially cells that are known as brown fat, sounds gross to me, but that's what those cells are known as. In some cells, additional membrane channels, look at the red circle on the diagram. That's an additional membrane channel. It's called UCP1 in this case. Allow hydrogen ions to pass through without making ATP, and then their energy that's being wasted, in essence, it's not being captured and being used in a chemical bond. In this case, it's just the energy is being released, and that energy gets released as heat, and it warms up the body of the organism. So uncoupling cell respiration, it's actually oxidative phosphorylation. Uncoupling that from the formation of ATP allows the organism to generate heat. Okay, and then just a couple, we're super close to the end. I think I will do the review at the end, even though this has been a long set of notes. You can always skip it if you want, but 2 more slides and then we'll do a review. I just want you to be aware that when we're learning, all you ever hear about is glucose. But there are all kinds of other foods. When you eat pizza, it's not just made of glucose. So how does that get turned into ATP? So what we're never going to learn, but you need to just see, is that fats, proteins, and carbohydrates can enter the process in different stages. So there's different sections where they enter the process. The carbohydrates enter at the beginning, fats enter a little bit later, proteins enter even later than that. So just be aware that they enter at different points. Okay, and then the other thing I want you to be aware of, you totally don't need to know a thing in this diagram, but you need to be able to interpret this diagram. So it says control of respiration. We don't necessarily want to be making ATP all the time at high levels of ATP. For example, if we're not exercising, we would rather be storing that energy instead of using up that energy to make ATP molecules. So we've got to control respiration. You've already learned about this process before, but I'm reminding you that it is in fact happening all over your body. Since every step, I'm reading the notes now, since every step of cell respiration is mediated by enzymes, there are multiple opportunities for feedback to control the process. So I want you to look at the diagram. I thought there was another bullet point. You don't need to memorize this, but you need to be able to understand what's happening. Let's just follow one of them along. So if you ignore everything and just follow the arrow, the white arrows going down, that's cell respiration. We're starting with glucose. We're cutting it up. There's an enzyme that's called phosphofructokinase. It's an important enzyme. You don't need to know the name of it. Just be aware of it. Okay, and as we go through the process, then you see the acetyl-CoA, you see the citric acid cycle, oxidative phosphorylation, and in the end, we make these ATP molecules. If those ATP molecules don't get used up immediately and they start, you start increasing them in the cell, you start having additional levels of them, they can actually serve to inhibit, change the shape of the phosphofructokinase so it stops working. Once levels of ATP drop off, then the ATP falls out of the allosteric site of the phosphofructokinase. It returns to its original conformation, and then it starts working again. So that's how this process can be turned on or off. If there's already too much ATP being made, then the ATP actually ends up causing the phosphofructokinase to change shape and shut down. You can see that there's the formation of citrate can do the same. It can inhibit the phosphofructokinase. And interestingly, AMP can actually trigger phosphofructokinase to start working again. AMP, if you can guess, is adenosine monophosphate. It's ATP that has actually lost two phosphates. So when AMP is present in the cell, it's an indicator that a bunch of phosphate has been used and it's time to start making ATP again. All right. So now if you want to be done, if you're so like, oh my gosh, I can't listen to her anymore, you could be done. I'm just going to go over, I'm reviewing what is it that you really need to know. Maybe this is a set of notes that you could come back to as you're getting ready for the test, but I'll just wrap it up here. All right. So things that I really need you to know ultimately when we're all done here. Number one, you need to know that cell respiration is a redox reaction. It's the reciprocal of photosynthesis. It is catabolic and exergonic. The energy that is released by the breakdown of glucose or other macromolecules is coupled, make sure you get that, remember it's releasing energy, it's coupled to oxidative phosphorylation. Phosphorylation means adding a phosphate that powers photosynthesis and powers the synthesis of ATP. Sorry, it powers the synthesis of ATP. So remember, ATP being made requires energy. It's endergonic. Cell respiration is exergonic. It's releasing that energy. If the process is not coupled, then heat gets released, and that's how endotherms like you and I generate our own body heat. Cell respiration in almost all organisms begins with glycolysis. The glucose is split to form 2 3-carbon molecules of pyruvate. A little bit of ATP gets made and NADH gets formed. And the pyruvate and the NADH are going to get moved into the mitochondria. However, sorry, I forgot this part. If no oxygen is available, remember that it catches the electrons at the end, and if it's not there, the whole process backs up. So if no oxygen is available, we go into fermentation. In animals, that's lactic acid fermentation, and it's a reversible process. Your muscles burn. Once you stop exercising and you start breathing and getting a lot of oxygen, the muscle burn goes away. It might last a few days even. And then yeast does ethyl alcohol fermentation, and it releases a little bit of carbon dioxide, and that's where those bubbles come from. All right, if oxygen is present, once we've broken the glucose into 2 pyruvates, we do the link reaction, which happens in the mitochondria. In a prokaryote, remember, it's happening in the folds of the membrane. The 3-pyruvate becomes a 2-carbon acetyl-CoA, and 1 carbon dioxide is released at this point. That's how we've lost a carbon in this point. And then even more, NADH is formed. And remember, that comes at the end. We really need those NADHs. Acetyl-CoA enters the citric acid cycle, and it leads to the production of even more carbon dioxide, more NADHs, and some FADH2s. Oops, and sorry, my picture's covering it, 1 ATP with every turn. Remember, there are 2 acetyl-CoAs that go through the process. It turns twice for 1 glucose molecule. The NADH and FADH2 then go to the cristae of the mitochondria, and they drop off their electrons in the electron transport chain. The energy that's stored in those electrons is reduced. That's the wrong word. I shouldn't say that. It is released. Reduced is a very specific term. And so it says the energy is released from the electrons, and it's used to pump the hydrogens into the intermembrane space, creating a proton gradient. The protons return to the matrix, that's called chemiosmosis, through ATP synthase, creating ATP from ADP and inorganic phosphate. That's oxidative phosphorylation. We oxidized glucose. We've been oxidizing glucose through this whole process. We've been removing electrons from it. They're being carried by NADHs. That's oxidation. And then we phosphorylate at the very end. We phosphorylate that ADP molecule to turn it into ATP. The electrons and hydrogen ions are taken up by the oxygen at the end then, which is called the terminal electron acceptor, because it's the final electron acceptor. The NAD8 plus and the FADs, those were electron acceptors also, but oxygen is considered the terminal electron acceptor. And so we form a little bit of water. If hydrogen ions are allowed to diffuse through other channels, we mentioned this already, uncoupling, the subsequent release of heat is used to warm endothermic organisms. And then finally, be aware that other macromolecules can be used instead of glucose. Glucose works through the entire process. Fats and proteins come in a little bit later in the process, and you don't need to know anything more about that. All right, ladies and gentlemen, that's it. Long set of notes. Sorry about that. Have a great rest of your day.